In this video, I'll be talking about the law of segregation, which is one of the most important and most basic concept of genetics. And as you all probably already know, that it was given by Gregor Mendel in the mid 1860s. So during the mid 1860s, Mendel conducted a series of experiments on pea plants, and one of which was a monohybrid cross setup. Now what he did in monohybrid cross was that he selected pea plants that differed in only one trait. For example, here we can see that there is a tall pea plant and another short pea plant. So they have all the traits same, and only the height of the plant are different. One is tall, another one is short. And he crossed these two plants, that is, he cross fertilized, and he observed that all the offspring resulting from this cross, they all were tall. So we will be calling this generation as the F1 generation. What he did next was that he self-fertilized these plants from the F1 generation, and he observed that in the next generation, which we will be calling as the F2 generation, he saw that there were both tall plants as well as short plants in a specific ratio. The ratio was 3:1. That is, for every short plant he observed 3 tall plants. Now from this we can see that the short trait disappeared in the F1 generation and it reappeared in the F2 generation. So whatever is causing the short trait must be present in the F1 generation as well, because otherwise how will it come in the F2 generation? So based on this experiment, Mendel concluded that every diploid individual must have 2 alleles. Now today we know these things as alleles, but what Mendel said was that each individual must have 2 genetic factors encoding a particular trait. Trait in this scenario is the height, and every individual plant must have 2 genetic factors responsible for the height of the plant. We now know that those genetic factors are nothing but alleles. So according to the law of segregation, Mendel said that each individual must have 2 alleles encoding a particular trait, and these 2 alleles separate or segregate during the gamete formation process, and 1 allele goes to each gamete. So now what Mendel did was he decided to designate these alleles or the genetic factors as alphabets. And as we can see over here, one of the trait, that is the tall trait, is persisting in the F1 generation, whereas the short disappeared. So the trait which is expressed in the F1 generation in the heterozygous state, that trait is the dominant trait, and the trait which is repressed in the heterozygous state is the recessive trait. So he decided to designate the dominant allele as a capital letter and the recessive allele as the lowercase letter. So here we can see that capital T small t represents the tall plant, homozygous tall plant, and the small t represent the recessive tall plant. Now according to law of segregation, when the alleles separate during gamete formation and the gametes is formed, each gamete will have 1 allele. Since in homozygous state there is the possibility of only 1 allele only, so all the gametes from the tall plant will have the capital T allele, and all the gametes from the small plant will have the small t allele. And in the F1 generation, although the phenotype, which is the observable trait, will be tall, but the genotype of the plant will be capital T small t, that is the heterozygous state. Now again, when self-fertilization is done in the F1 generation, again following the law of segregation, when the alleles separate during gamete formation, in this case we have 2 possible scenarios. The gametes can be either capital T or small t, and when the fertilization is done, we can have 4 possible genotypes, as we can see from this picture here. And among the 4 possible genotypes, 3 of them will give tall phenotype, that is the observable trait, the trait that we can see. So the genotype, we have 4 genotype, but we have 3 tall plants and 1 short plant. So this is basically what the law of segregation is. Now, when Mendel conducted these experiments, he did not know that these genetic factors are alleles. Today, we know that alleles are basically the forms, the different forms of the same gene. So, today we know that the traits that Mendel was studying was being governed by a specific gene at one locus, and alleles were the different form of the same gene. So, this is all about law of segregation.